you start with the brain being the most metabolically active organ in the body. Okay. And you focus on the single most important regulator of brain functions, and that's insulin. And secondly, you act to correct resistance to this regulator, okay, which is inherent in AD, the insulin resistance, and precedes AD symptoms. And third, you want to use a systems biology approach. Don't go after one-off pathology-specific targets. They haven't worked. Use a pluripotential drug target that can bypass the multiple dysfunctional insulin signaling pathways. And so 10 years ago, seeing that insulin resistance was really a key component in this disease, making Alzheimer's disease a metabolic disease in which the brain is just getting starved of the energy it needs. And there was a drug I was working on at another company for type 2 diabetes. That great drug, excellent pharmaceutical properties, excellent safety. And I coupled that this company that I had left was going to change business models. And so this great drug was going to be sitting on the shelf. And I was putting two and two together, and it came up to 20 that just this drug could actually be a great treatment for Alzheimer's disease. Key in that was looking at the target of our drug is a nuclear receptor called PPAR delta. It also agonizes in a secondary way another nuclear receptor, PPAR gamma. But when you knock out the gene for PPAR delta, these mice get so many attributes of AD that it was just making a compelling story that, oh, this drug could, because it hits PPAR delta and can really be an effective treatment for Alzheimer's disease. So that was really the start of things. The rationale for that Phase IIa study design, was that here we are, a startup company with a limited budget. We have limited time. Time is money. We had limited GLP toxicology covers. Therefore, we could only dose a human being at that time for 2 weeks. We had a limited supply of GMP drug product. And we didn't want a study in which we would measure CSF, because that was an undue patient burden to have 2 lumbar punctures in 2 weeks. So we designed a study that first explored safety. This was the first experience with this drug in elderly, quote unquote, compromised patients with this new chemical entity. Second, we wanted to examine the widest possible range of doses to find safe doses. Third, we wanted to examine multiple indicators of potential target engagement and efficacy to find one or more positive indicators because of the unprecedented early time, 2 to 3 weeks to take outcome measures. And we wanted to explore the potential to potentially identify drug responders. And lastly, fifth, enable rapid recruitment and completion to effectively compete for patients with other longer AD trials. So we did such a study in 36 subjects. And what we attained from that was multiple indicators of potential improvement in ADAS-Cog and in other measures, and looking at cerebral glucose metabolism via FDG-PET. We saw major changes in brain glucose metabolism that were aligned with the potential to improve the dysfunctional glucose metabolism inherent in the disease. One of the important points here is that the field, there was a third pathological hallmark of the disease noted by Alzheimer in 1906, and he called them adipose inclusions, or fat deposits that was observed within cells. And that's been ignored largely. And that is very important. I mean, the brain is half lipid, and so lipids are vital for structure and function. And so this drug addresses that. I think in 2015, these adipose inclusions were thought to be largely triglycerides. And so our drug has a mechanism of action in which we've shown in animal models of diabetes and so on that we reduce triglycerides to a significant extent. So that could address that third pathological hallmark as well.